cause liver cancer. Okay? So instead they are caused by lifestyle factors, toxins, and gene mutations. Examples of these diseases are diabetes, cancer, other immune diseases such as psoriasis, etc. Now, pathogens and disease. So pathogens are the microbes that are capable of causing tissue damage. Alright? It's fine if they can live in our body without causing destruction. Okay? What are the examples of microbes that live in our body that doesn't cause destruction?
E. coli?
No, E. coli will give you diarrhea.
By live in your intestine.
Yeah, you have things like lactobacillus, so there are things like probiotics. Okay, so you have your good bacteria and your bad bacteria, and there are also some pathogens or not pathogens, there are even some microbes or microorganisms that don't do anything. They're not beneficial to you, and they will still also won't cause you any problem. Okay. And pathogens be the bacteria or not. So there are 4 types of main pathogens. They are the viruses, bacteria, parasites, and fungus. Alright? So the key features of immunity against an infection is basically number 1, we've got the defense against the infection, which is firstly mediated by the innate immunity, followed by the acquired immunity. Alright? So the innate immunity represents the first line of defense via pathogen associated pathogens. This is what I mentioned here the other day. And then you've got the acquired immunity that represents the follow-up of the defense based on the innate immunity generated. Therefore, if the pathogen evades the immune system, you will be able to replicate even further to cause more damage. Okay? So let's start with the first one, viruses. Okay, so this one, the first image there, we see until we're very tired of it. That's the coronavirus. Alright, and then you've got other viruses such as HIV, hepatitis B, Ebola virus, adenovirus, influenza, bacteriophages. Alright, but bacteriophages doesn't actually infect us. Alright, it basically infects bacteria, but these are the different types of viruses available. So the thing is that viruses are obligatory pathogens. Now what does obligatory pathogens mean? It basically means that they are an organism that requires the replication mechanism of its host to cause damage. Okay? It is divided into 2 main categories, which are the DNA and the RNA virus. Now virus can also be subdivided into the enveloped and non-enveloped viruses. Okay? So if you look at this illustration here, you'll be able to see that at the top you've got your DNA viruses. Okay, they could be enveloped and non-enveloped. Envelope meaning here they have an envelope that encircles their capsid. Alright, and then you've got your RNA as well. So RNA is a little bit special, whereby you've got your single-stranded RNA and your double-stranded RNA. So for single-stranded RNA viruses, most of them are enveloped, but for double-stranded for non-enveloped ones, you have the double-stranded as well as your single-stranded. And those are the key viruses that is available. However, I am not here to lecture you all on the different diseases. I'm here to tell you all about how it functions with the immune system. So you do not really need to go and remember all these names. Okay? This is unnecessary. You just need to know what are the main categories. Okay? Now, the thing is that if you look at this flowchart here, you see that double-stranded RNA virus basically enters into a cell, okay, whereby it will involve RNA synthesis to convert it into more RNA, and then you have your translation, and then your translation, and then you will have your RNA synthesis together with the formation of new virus particle. And this involves packaging, because part of the RNA will be used as the core data to be packaged, and then the okay, so virus is basically just made up of DNA and also protein. Okay, so that is for double-stranded RNA. For positive-strand RNA, you basically don't need to go through the RNA synthesis because the strand is already similar to your mRNA, whereby it will directly translate. Alright, so this one enters, it will basically translate, make more protein virions, and then it will be packaged in order to make more viruses. And last but not least, you have retroviruses, which are normally DNA virus, and this DNA virus requires something called reverse transcription, okay, whereby then it will produce more of its so basically what's going to happen is that the reverse transcription will then lead to DNA integration, and this DNA integration means that it could integrate directly into the genome of the cell, okay, and based on that integration, they will then use the host transcription and translation system to make RNA, to translate, and then to package to make more viruses. Okay, so that's why it's called an obligatory pathogen, whereby it requires the host mechanisms. But virus is not really alive and not really dead. So it's a combination of both, whereby it just requires something to allow for it to propagate.
So is prion also born from this thing?
Nope. What are prions?
Only proteins.
Sorry?
Only protein.
Yes. Prions are only proteins. So prions are different categories, and prions basically are the proteins that causes toxicity, or they have certain enzymes that leads to certain pathways that cause damage to the cell. Okay, so prions are very similar to toxin. It's just that toxin could be chemical structures. Okay, but prions are proteins. Okay. So the thing with viruses is that there is two pathways. They have the lytic pathway and also the lysogenic pathway. Now, when you see the word lytic, which means that it lyse, okay, and then the other one lysogenic, it doesn't lyse. Okay, so when a virus is infecting a cell, okay, it will basically, for example, let's start here. Yep. So it basically infects the cell, it goes into the cycle whereby it makes more of its DNA, makes more of its protein, repopulates and repackage. Once everything is done, it will break open the cells. So this is what is causing the cell damage. Okay. So once cell damage occurs, okay, they will release out to infect more cells. Right. So this is the lytic pathway. However, there are also situations whereby it goes through the lysogenic pathway, whereby what it does is that it will just replicate more and more of its own DNA within the cells. And when that happens, it will just keep propagating and propagating and propagating until one day it feels that it's ready, then it will move into the lytic cycle to cause disease. So during the lysogenic pathway, it's normally in a dormant state, which means most likely there is no symptoms of a disease, sort of like incubation. Alright, so these are the two pathways that viruses take. So now, what we want to understand more is that how does our immune system actually act against viruses? Okay, it starts with the innate immunity, whereby it's a direct stimulation of interferons. Okay, whereby these interferons are antiviral protein or glycoproteins that are produced to counter the viral infection. Okay, so you got interferon alpha that is made by leukocytes. You have got interferon fibroblasts, and you have interferon gamma made by NK cells. Now, aside from just using interferons as an defense against virus, you also have NK cells. So what happens with NK cells is that they will basically kill or lyse cells which are infected by viruses. Okay? So they're basically signals that actually allow for the NK cells to recognize that a certain cell is basically being kill them off. Okay? What about acquired immunity towards virus? Well, the key ones would be the humoral immunity that involves antibody, which are specific for viruses, which can be stimulated for viral clearance. Okay? This is very similar to what we have for COVID, whereby you inject the vaccine and then you produce antibodies. What the antibodies does is virus particles. Now, why is it that at the end of the day, some people still mention that the injected COVID vaccine was not very, it's good for preventing death, but it actually doesn't stop you from getting COVID, right? Anybody here got COVID before? COVID. Who? Okay, the easy answer is who haven't got COVID before? No one. Sure, you didn't even have a flu or anything. Serious?
Yes.
Okay. So you're the only person then. That's because I got COVID. Dr. Venu just got COVID for the second time. So yeah, why is it that even with the vaccination, we're not able to stop COVID from infecting you?
COVID has gone straight.
No. Why?
So the like variation of the genetic of the virus.
That's what the answer.
Yeah.
No. Why is it that we
the
can't?
development of the immunity of the depend of the immunity of that person, because some of the people like cannot, their immunity not working very well.
No, there is 10 of us in this class. I think most of you all have got the COVID vaccine, right? And only one person didn't get COVID. Why?
Mutation virus?
No, nothing to do with mutation of the virus. Okay, where did you get your vaccination at? That means not from what country. Where was the vaccine administered to you on your body? Muscular, right? Okay. So when it's given through your muscular, what's going to happen is that you're going to generate antibodies that is circulating in the body. Okay. Where does COVID enter your body through? Nose and mouth. Okay. So if you have some level of understanding of immunity, say got understanding it, right? Okay. So why is it that the antibodies that are generated within the body cannot reach the nose, but it could basically stop you from getting killed by COVID? I mean, out of 10%, you're still alive. Although you got out of 9%, you got COVID, but you're still alive. Okay. So why?
We already have.
As you said, like the antibody that so but we the bloodstream. But for most part, and I know, but the virus is not infecting or entering the cell in that case.
No.
The virus does not.
Very close, but not really.
The virus does not affect cells.
No, it affects the cell. The virus you don't interact with the cells.
Affect the cell with no like.
Because
no
look, what's happening in that time, like we are getting virus infected by virus. The first of all, it's innate immunity, so we have symptoms because of the innate immunity.
No.
I'm sorry, like this.
No. Yes, it's a good try. It's a good try. Anybody else want to try? Okay, so this is where a much better understanding of immunity comes in. The antibodies that's being generated in the body is called known as IgG1. IgG1. Okay? So it's what? Immunoglobulin complex 1. Okay? Now, in our nose, okay?
We have HA.
Yes. In the nose, we have IgA, respiratory system. Yeah? IgA. Alright? IgG cannot go into IgA's area and fight with the virus. No, you didn't say IgG and IgA.
Okay, I know, but I said it's not
That's why I
necessary
said it's
to
close. I didn't say you're wrong.
Yeah, I know.
Alright? So the thing is that because of how the respiratory system works, it requires something called IgA, immunoglobulin complex A. Alright? So the IgA in the nose is the one that actually is going to stop the virus from, it's going to block the virus or cause the virus to flee. Okay? But once it enters into the nose without the IgA and through the respiratory system, it will start infecting and start breaking cells. That's why we start to have symptoms of COVID. Okay? It's just that the vaccination basically stops further destruction of our lungs. Okay? Because when somebody gets COVID, basically what's going to happen is that they will start to have alveolar cells breaking up. Okay? Then you're going to have fluid building up in the lungs. Okay? End up with pneumonia, and then you got multiple organ function being shut down because there's insufficient. IgA, sorry. Okay, so that's why nowadays a lot of people are actually working on respiratory vaccination through the nose. Okay, I think in China you have smell right? Yeah. Which university? Xiamen University. Yeah. Heard of Biotech before? Yeah. So Biotech is actually also developing that. And then all around the world, US, they developed the mRNA vaccine through the nose as well, nasal spray. Okay? I think the most special one will be the one from China because it's a cup, it's sealed. They basically put a gas into the cup. The person open, goes to the next door. After they collect the cup, he goes next door, open up, and then they just inhale everything in. Okay? Alright? But these are all newer technologies that's been developed because for you to make or encapsulate the virus of the vaccine, for it to be capable of reaching the nose, would have to fulfill certain things. Okay? It has to be small enough, it has to be, there's a lot of things that will happen. Alright? So that's why for COVID, you can still get COVID even if you're vaccinated, but if you take those small ones or the nasal vaccine, the chances of getting COVID is much lower. I think protection against 80%, 90%. Okay? So that is why it's important to understand what is happening. Alright? So that will be tomorrow. Now it's also important to clear out viruses before they attack cells and reside in it, so which basically causes acute infection. So this is what happens when in the humoral part of the body, which means in circulation. Anywhere in the bloodstream, anywhere in the lymphatic circulation. Okay? So, if you have a look at this very simple graph, this is how antibody works against your virus. So take for example HIV. Alright? So you've got the neutralization phase whereby the antibody neutralizes the antibody by blocking its ligand or its sort of something like a spike protein from attaching to the cells. Okay? So you have neutralization whereby it blocks. Then you have complement lysis whereby you have antibodies that come and then they recruit something called a complement, and then the complement basically cause destruction to this. Take it as this as the targeting system, this as the payload or the killer. Okay? You also have opsonization and phagocytosis, whereby your antibody will then trigger Fc receptors and also complement receptors, which basically lead to phagocytosis. And then you also got antibody, whereby it's normally going to be a killer NK cell, and this NK cell with the Fc receptor will bind to the antibody that leads to the killing of the virus. Okay? So this is how the humoral immunity works for a virus infection. Okay? Sometimes even at the infected cells, if the antigens are being presented on the cell surface, antibodies can still come and attack. However, if they do not present any of this, so take a guess, okay? What sort of virus would actually lead to your antigen being presented on the cell surface?
What sort of virus?
Yeah. So based on, for example, this. What sort of virus would basically lead to something being presented on the cell surface? Have a look at A, B, and C. Take a guess, which one would be the one that would actually allow?
The neutral virus?
Why?
Because it leads to
I'm giving marks. It will be 1 mark for the correct answer, 4 marks for the explanation.
What's the question?
So the question is,
Why you think retrovirus is the one?
the question is, okay, this is an infected cell, okay, but there will be virus particle being expressed on the cell surface, so the antibody can come and bind to this infected cell to kill this cell. Okay, so I was asking, out of these three, which one will actually cause that sort of answer?
Because the envelope will be like emerged with the cell membrane.
No, that's what the answer is.
Oh.
No,
And the other one, like the cell, it will release interferon and
No.
And the viral, it will come out. That's right.
no. Take a guess. Why? What is the difference between A, B, and C?
This one, like
The
it will control
Integration.
integration of what?
DNA integration into the genome. Alright? When there's integration of the protein in the genome, DNA into the genome, okay, it will constantly produce the same protein and then being able to be expressed on the cell surface. Alright? And when that happens, you can then trigger this. Does not as well, but this does. Right? So only when integration into the host genome occurs, you will be able to trigger that sort of the reaction at the bottom, whereby an antibody can detect an infected cell. Because on a normal standard immune reaction, your antibody don't go and bind to infected cells. Okay? Normally. Alright? Your T cells do that. If you understand what Dr. Liao explained for acquired immunity. Alright? So then, for acquired immunity, okay? So, cellular immunity is important when chronic infection is established. Okay, what is the difference between acute infection and chronic infection?
Acute means slowly growing infection will give you
No. Who said very fast now? Very fast?
growing infection.
Sorry?
Speed or long infection.
Speed or long infection is what? Acute or chronic?
No.
I'm asking what is acute infection? Acute is fast onset.
It's the first onset of the
Okay. So an acute infection is an infection that occurs when you quickly get the symptom, but after a few days the symptom disappears. A chronic infection is an infection that occurs and then constantly persists over a long period of time. Alright? So your acute infection is normally cleared off by your humoral immunity. Look at that, acute infection. And your cellular infection immunity is important when you have a chronic infection. Why? Why? No, why is it that cellular immunity is important when you have a chronic infection? What happens in a chronic infection? It means that the bacteria or the virus persists in the body. Okay? But if you're under attack all the time, you need to find somewhere to hide. Correct? You need to find somewhere to hide. A cellular immunity is important when the viruses are hiding inside the cells. Okay? And normally cells, when viruses, when they hide in the cells, you have a chronic infection. Why? Because they are protected from any antibody attack. Okay? So when they keep staying in the cell, they can make more and more and more and more of themselves. Alright? So when they're hidden, that is where the cellular. Okay? This is when the virus decides and starts to replicate in the cell. Okay? This is crucial for disease clearance and it normally involves CD8 T cells and CD4 T cells. Okay? Now, just quickly share with you all. This is a virally infected cells. Okay? You see virus particles, everything inside here. Alright? Now the thing is that although it doesn't goes out and sit on the cell surface, like what happened here, okay, you can see that the virus particles are all inside the proteins. Okay. However, the human body or the cells, most of our cells, have a very crucial thing called what does MHC1 do? They present. They present antigen to the cellular immunity. Okay? Why? Because cellular immunity, which involves CD4 T cells and CD8 T cells, have something called immune surveillance. Okay? B cells or antibody cannot do immune surveillance. So what is immune surveillance? Immune surveillance is being capable of going, checking each and every one of the cells to see if they are normal or abnormal. So, once your MHC class 1 complex carries a small part of the virus particle, it will then be presented to the T cells. And what's going to happen? The T cell will identify that particular peptide sequence 9, 8, 10 mers to be something that it doesn't recognize. It is not in his memory that they have seen this before. So what is going to happen then? It will basically lead to. So actually you start here, sorry. So you start here. So you got your virus infection, then after that you have all this thing, then present it to the naive T cell. And then once this is presented to the naive T cell, and the naive T cell said that I didn't see this before. When I didn't see this before, it means that it is going to be something foreign. And because it's something foreign, before I go a little bit further, why is it that our immune cells don't attack our body?
Because our cell have a normal MHC.
Well, it also has a peptide on it.
So it recognizes that peptide.
Okay, so how does it know that it is ours?
Yeah, like, do you train when you are in the
What?
virus?
In the timers? No. See, you guys are getting very close. So, why is it that our body's own cells don't attack our CD8 T cells or whatever, recognize that this is your own cell, no virus infection, no cancer, and they don't attack? Why? So, she says that it's been trained in the timers, trained correct, but not in the timers. She said what? What did you say just now?
It's trained before.
It's trained before. Actually, it's nothing to do with training.
Maybe it's search.
Ready? Okay. Let me give you a quick tip. It happens before you were born. It happens before you were born. I like this type of classes. I like this ask questions. No, actually it's easier to learn when I give you questions.
Correct.
Because you remember it better. Because when you just read, you won't remember the importance of it. But when I keep asking questions, then you guys will, you know?
I can recall.
So why? It's all cheating, really.
Cheating.
Okay, so the tip was that before you were born. Okay. Now, the human immune system, okay, when you were a baby, when you were a fetus, okay, the immune system will start to eliminate. Okay, clones of T cells and B cells that binds to any of our cells. Okay, which means that okay, I don't know whether you all know or not, but there's something called clonality. The immune system, for example, a B cell, each antibody is different when it binds to different thing. Okay? Now, how do you get that many clones? It's because the human body's immune repertoire is between 10 to the 12 to 10 to the 50 different clones. Okay? It's predicted now. Alright? With that big range of clonality, okay, you are then able to target any antigens that come to the body. Be it for B cell or for T cell. Now T cell, the variation occurs at the B cell receptor. Okay, that's what is used to recognize the antigen being presented by the MHC class 1. Okay. Now the thing is that before we were born, when we were in fetal stage, okay, all of those clones or all of those T cell receptors or B cell receptors that can bind to our own antigens will then, okay, that means you kill off all those cells, okay, your body, all your bodies would have killed out all the cells, okay, T cells and B cells that could recognize your own antigens. Okay? And when that happens, okay, you will then, before you were born, you have gotten rid of anything that recognizes your own body. When you are born, that is when any binding will not be destroyed anymore, but it will go into a process whereby it will differentiate and proliferate.
Sorry, how you
No!
not totally.
Okay. Imagine this is just one of the clones.
Okay.
This is your own body's, this one. Okay. Now you have many of these. Alright? But what it binds to is different. So anything that binds to your own antigen, okay, this original cell will be deleted.
Clone that bind into the human antigen.
No,
Human
your
cells.
antigen.
Yeah.
Your antigen will be
It will
deleted.
be deleted the cell.
Yes.
Okay.
So the whole cell will be deleted. That means the DNA sequence that carries the receptor that recognizes this will be deleted.
Okay.
So what about like other cell like we have in the cells in our brain there?
Everything, all your cells. So the immune system
So
during that
how
time will go and target. What's going to happen is like it's sort of like a negative selection, whereby it's not called negative selection. Basically, it's called clonal deletion. Yeah, clonal deletion during fetus development. So you can go and look it up.
Yes.
Alright? So what's going to happen is that you will all kill. So all the cells or all the clones that can bind to any of your cell in the body, brain cell, skin cell, bone cell, whatever cell, it will all be deleted. And what is left are things that don't recognize at all. Okay? So when you are born with an immune system, okay, that don't recognize, that do not have anything that will target your own body.
So what about like where is that clone coming that attack
That
human cells?
that is the clone.
Oh,
that
there's
is the
some.
clone. That's the clone. Because like I said, the human immune repertoire is I cannot count about that. Okay? It's 10 to the power 15 clones. Okay? So wait, billion is 9, trillion is 12, yeah, so it's more than trillion. Okay? So all that will be deleted. So when you are born, okay, when you are born from your parent, your mom, okay, the immune system then turns into a switch on mode. That was the fail-safe mode where things are being deleted. Once you're born, okay, that reaction don't occur anymore. Killing of immune cells don't happen anymore. That is when your immune cells will start to be alert. Okay, because you're out of the protection of your mother's womb. You are now faced with the reality of the world with pathogens and everything. Okay? So that is why the human immune system doesn't attack the own body. It's because there is no clones that can recognize our body anymore. All of them has been deleted. However, what about autoimmune disease? Okay, let's stop here. Autoimmune disease, we continue in the next chapter. Alright? So, There is also cases where you have evasion of the immune mechanisms by virus. Okay, just like how we are talking about cancer, everybody heard that cancer there's also escape of the immune system, right? So viruses can also escape. So how they do it? So viruses have evolved numerous mechanisms for evading the host immunity. Okay, so a number of viruses have strategies to evade things such as complement-mediated destruction. Okay, so you can see there's some what you call it graphical there, but this is not important. The next one is the most important one. So virus will also escape immune attacks by changing their antigens. Okay, this is what happens with. How do they change their antigens? Yes, by mutation. Just like what happened to COVID. Okay, so when our body adapts, okay, to that particular antigen, all of them will be wiped out. However, viruses are very prone to mutation. They can mutate very easily. How do they mutate? How do viruses mutate? They cannot even make new cells, new virus. How do they mutate? Sorry? How does that happen? Right. Okay. There are also certain instances where they basically go into the human genome, and then certain parts of it will basically be changed. There are also things like transposons. Okay, so viruses do have transposons, and what's going to happen is that they will basically change their genetic sequence. Now the thing is that if they keep changing their genetic sequence, don't they die? If, for example, this genetic sequence changed the spike protein, and the spike protein becomes non-functional anymore, will they die? No.
But they cannot infect another cells.
Yeah, if they cannot
But
infect the cells,
sometimes
they will
like they
still.
will infect more type of the cells.
But the thing is that they cannot infect the cells. How are they going to grow?
They cannot infect.
Not that you die.
Exactly, because they can't infect other cells, they'll die.
We hope.
And when they die, you can't find them.
Yeah.
Can you find them if they die?
No.
No. Okay, because they don't make viruses which can infect a person. Right? And if the virus cannot infect a person, will it be detected? No. If you're healthy, you don't just go to, I want to go do a COVID test. No, right? Correct? So that's why whenever we get reports of mutation, it's always mutation which leads to worse or stronger infection or more virulent strain. Okay? Why? Because the normal strain has been contained by the immune system, and then whatever that can escape, okay, will tend to be more virulent. But only those which are functional can escape. Because if they are not functional, they can't escape. And when they cannot escape, what's going to happen? That time they were predicting within 2 to 3 years. Okay, because of how fast they mutate. Every 2-3 months you get Omicron, and then after Omicron 4B, 2A, whatever. Now it's what? I don't know what called already. I also cannot remember
Maybe they
anymore.
died.
Sorry?
Maybe they died.
No, it's still there.
Yeah, I know.
Yeah, it's just that our immune system is adapting to it. It's just like bird flu, H1N1, MERS. So the immune system starts to get accustomed to it, adapt to it. It becomes less severe. Okay. So a large number of the viruses can also evade the immune response by causing generalized immune suppression. Okay. So if you notice here, so this is basically an immature DC, And then what's going to happen is that, oh, this is on tumor antigen. Don't bother with that. Yeah, but mutates. Okay, and when it mutates, your immune system is finite. Your immune system is finite. The repertoire is finite. Okay, so once you are facing this infection, it becomes a lot of the same army to fight. However, if the virus changes its antigen, this army will have to disappear first before a new army can be brought up to fight. So that's why you get escape. Okay, so that's what happened when you have immune evasion of the virus. Okay, all right. So that's 45 minutes. Okay, so shall we take a quick 5 minutes break so that you all can go toilet or whatever, or you all don't need a break? Need a break? Need a break? No. Okay, they're really enjoying stuff. Okay, next, bacteria. It's very good. I talk too much. No, you guys, I force you guys to talk a lot. So, immunity towards bacterial infection. Now, immunity to bacterial infection is achieved by means of antibody, unless the bacteria are capable of intracellular growth. The two types of bacterial infection are extracellular. Extra means outside. Cellular means cell, so outside of cell. Intracellular means inside cellular cells. Okay. So if you have a look at this is an example, okay, of extracellular and intracellular infection, and then you have got the different organism. But this is how it normally happens. So for extracellular infection, the sites of infections are normally interstitial spaces, blood, and the lymphocytic lymphatic system, or it could also be on epithelial surfaces. Okay, which means if it's extracellular, it has to be outside of the cell. So if it's outside of the cell, where do they be? So they will be on your skin, in the gut layer, outside of the layers. It could be in active. So what happened to all this TB? So the thing is that the TB basically can enter into cells, okay, and form a protection layer called a granuloma and goes into hibernation. This normally occurs when the patient or the infected person has good immunity, because your immune system will try to get rid of all the bacteria. But in order for the bacteria to save itself, it infects a cell and hides in the cell. Okay, this normally happens with TB. Okay, and it basically resides in macrophages and alveolar cells. Okay, now the thing is that the statistics is that 33% of the world population has latent TB. But is it going to be something which is bad? Or when do you think it will? Do you think that it will be a bad thing that somebody has got latent TB? Not sure. The good thing is that you won't have any signs of TB. The bad news is that if you have diseases such as diabetes, you have cancer and require immunosuppression, if you need to undergo GVHD, you need to undergo transplantation. Okay, these are the cases when the body's immune system will drop. Okay, actually during COVID, we also studied correlation between TB and also COVID, because COVID it doesn't actually lower your immune system, it forces your immune system to focus on one infection. and then we call it a dual infection, whereby the latent TB comes up. Alright? So when the immune system is down, TB starts to come out. Okay? So this only will occur if there is an intracellular infection. Okay? Now, how do you actually get rid of an extracellular and intracellular infection? So for extracellular, you basically have complement, phagocytosis, antibodies, antimicrobial peptides, etc. If it's cytoplasmic, you need NK cells and T cells. For vesicular, also the same, T cells and NK cells, but they depend on macrophage activation. Okay?
Means that macrophage has to be ABC.
Yeah, your macrophage will have to be the one that is presenting the antigen to the T cells. That then will lead to this one. Why macrophage is important? Because the macrophage can break down things. If they are in the cytoplasmic area, it's very easy for the MHC molecule to take it up and put it outside. But if it's in a vesicle, okay, your MHC class 1 molecule cannot reach it. Okay? So you need macrophage to eat them up, chop them up, and then present them, only they will be able to recognize. Alright? So, extracellular bacteria are capable of replicating outside the host cell. They are caused by two principal mechanisms, whereby many of these bacteria produces toxin. It could be endotoxin, it could be exotoxin. Alright? So the immune response against extracellular bacteria are aimed at eliminating the bacteria as well as neutralizing toxin. Okay? You need to also neutralize the toxin to stop any further damage. Because it's the toxin that actually causes the damage, not the bacteria themselves. Okay? That's what they secrete. That's why they are good bacteria and bad bacteria. Your good bacteria helps to, example, bacteria in the stomach helps you to break down food that you cannot digest. Alright? While the bad bacteria will basically produce toxins to damage your cells. Alright?
But the toxin wasn't purposely induced, just like
Yeah, it's not.
side product of their metabolism.
Correct. Ever heard of the word, what's the phrase? One man's meat is another man's poison. One man's meat is another man's poison. Heard before?
One man's apple is another man's.
I don't know. I always use one.
I know I use pepper apple.
Anyway, take this for example. Let me think. Okay, soybean milk, soy milk, 豆奶，豆浆，豆浆。Okay, so soybean has got a particular toxin when you drink them. Okay.
I drink a lot.
There is a particular toxin. Okay. Now some people who have stomach discomfort and everything, they can't take that toxin. Okay. But some people they can. Alright. So then you have this fermentation method, for example, tempeh, Malaysian tempeh. You have the Japanese natto. You have the Chinese doujiang. What's that? The fermented beans. No, tofu is not. No. The beans in the black paste. Alright? So, different thing, for example, it's not purposely that the bacteria produces something that is purposely to kill us. It's just that is their byproduct in their genetic system. Okay? But the good bacteria, like your lactobacillus and everything that you take from cultured drink and everything, okay, it's because they produce certain enzymes that help you with digestion. It's also something they produce. Okay? So it's not purposely that they are aimed at killing us, it's just that is their byproduct. Okay? And those are the byproducts that will actually harm us or help us. Okay? So the immune response is aimed at eliminating that. Alright? So you also got innate immunity towards extracellular bacteria by phagocytosis, by neutrophils, monocytes, as well as tissue macrophages. And you also have activation of the complement system in the absence of antibody. So with antibody, you can recruit complement, but sometimes you don't have antibody, so it will basically have the complement system to attack. Alright? In terms of acquired immunity towards the extracellular bacteria, humoral immunity is the principal specific immune response against them. Okay, so you have things like strong that are caused by the polysaccharides or the sugar from the bacteria, and antibodies such as IgM and IgG also act against the bacterial surface antigen, as well as a toxin that stimulates 3 different types of effector mechanisms. Okay, so it could be your innate, which is caused by all these things, alright? And then you have your acquired, okay, which is most likely in the form of antibodies. Alright? So what is the main mechanism of action? Okay, so you have your IgG antibodies that opsonize the bacteria and enhances phagocytosis, causing the ingestion by macrophages. Okay, which means what it does is that all target for your macrophages, and then your macrophages will just go and ingest that. Okay, but of course there is also Fc region or Fc receptors on the macrophages that binds to the antibody that allows for opsonization to occur. Phagocytosis to occur. Okay, so that is opsonization. Now, antibodies can also be used to neutralize the bacterial toxin. Okay, and your IgM and IgG antibodies activate the complement system as well. Where your complement system will be activated, you have things like the membrane attack complex and everything, which sort of breaks the extracellular bacteria. Alright? Now, in terms of the immune evasion of extracellular bacteria, okay, notice there is no T cell responses.
Yeah.
Why there's no T cell responses?
Because it's extracellular.
Exactly, it's not in the cell. If it's in the cell, then you call your cellular immunity. If it's not in the cell, although a bacteria is a cell, but you will still require antibody to get rid of that. Okay? So, immune evasion of extracellular bacteria. So, because of genetic variation on the surface of the antigen, okay, that is one of the ways that the bacteria can use to evade this specific immunity. Like I said, you generate a whole bunch of antibodies that target a specific antigen. The bacteria can basically just change its surface antigen. Alright? How does it change its surface antigen? Is that it might go through several phases of condition, just like mycobacterium. Instead of, so the mycobacterium hides in the cell, but this one it doesn't hide. Maybe it just changed the state of the bacteria. So maybe from an active stage into a passive, whereby instead of producing gram-negative and gram-positive bacteria also contain one or more sialic acid residue, and this sialic acid residue basically inhibits the complement activation by the alternative pathway of complement activation. I think Dr. Liao will teach complement activation, right? Your membrane attack complex and everything. Correct? Yeah, so you should understand what is the alternative pathway and what's the conventional pathway. If you don't have, never mind, you can go to find out. I say I want to share with you all the videos where I haven't shared. Never mind, I'll share it after today.
What's the
Send the text to remind you. Sorry?
What's the capsule?
Capsule? What is encapsulating in an outside? Where they are inaccessible to circulating antibodies. Alright? So the elimination of intracellular bacteria require immune responses that are very different, okay? Whereby you basically have something different. So in terms of innate immunity towards intracellular bacteria, during the innate immune response to intracellular bacteria, phagocytes ingest and attempts to destroy the bacteria. Okay? Now, intracellular bacteria are resistant to degradation within the phagocytes. Okay? So this intracellular bacteria can also activate NK cells either directly or by stimulation of macrophages to produce IL-12, which is a powerful NK cell activating cytokine. Right? So in terms of innate, it's normally phagocytosis, but sometimes it's resistant. When it's resistant, you need NK cells to come in. And NK cells will only help when there is good amount of IL-12 being produced. Right? Now, in terms of acquired immunity towards intracellular bacteria, you can only look at T cell responses. Okay, so the cell-mediated immune response is the major specific immune response against the intracellular bacteria. There are two types of cell-mediated reaction. Number one is killing of the phagocytosized intracellular bacteria as a result of macrophage activation by T cells, which is derived of cytokines, particularly interferon gamma. So you basically require interferon gamma to actually cause the destruction of the cell. Or it could be lysis of the infected cells by cytotoxic T lymphocytes. Okay? So there are two methods lah. Now, in terms of evasion of immunity by intracellular bacteria, as we all know, because it's really go into the cell, it will have methods or ways to basically stop phagocytosis from happening. Okay? Some intracellular bacteria also do this by inhibiting the phagolysosomes fusion, okay, which means that it blocks the phagolysosome from working. Or it could produce something called hemolysin that blocks bacterial killing in macrophages. So if you look up hemolysin E, okay, you'll find that it's crucial for things like Salmonella typhi. Okay, so Salmonella typhi can basically be reside by using hemolysin E to block bacterial killing in the macrophage. So a lot of people are using hemolysin E as a method for detection of intracellular Salmonella typhi infection. Alright? So the thing is that when you understand all of these things, right, and you think of, how do I come up with a detection method for Salmonella typhi, for example, you need to really go and study and understand the mechanism of the bacteria. You need to understand it better before you're able to come up with something. Same for the immunotherapy as well. Alright? Imagine if you're trying to use antibody to kill an intracellular bacteria. Don't make sense at all. Okay? It's the same thing when last time during COVID, Donald Trump was given antibodies from infected patients that recovered. Sorry? Yeah, convalescent plasma. Yeah, he was given that to treat his COVID. Okay, doesn't really work that well. It would basically stop the virus from happening. By the end of the day, you need to get rid of the infection which is within the cell as well. Okay, you need to destroy the source. That one basically just helps to reduce the symptoms and doesn't actually help to clear if his immune system was not good enough. Alright? Toilet anybody? No? Still okay, huh?
Okay.
Okay, huh? Anybody need? No? Okay. I've got the most. I need to drink more water. Okay, next part. Fungus. So, what are fungus? Fungal infections are eukaryote that tend to cause serious infections, primarily in individuals with intact immunity. Okay, this is a very important line. Okay. The important line here is serious infection, impact immunity. What does impact immunity means? Weak immunocompromised, weak immune system, and etc. Okay. A normal person don't normally get fungal infection. Okay. A normal healthy person don't get fungal infection. It will only occur when the person has that impact immunity. Okay. So you can see there are things like this pityriasis versicolor. Okay. You got feet fungus and you got thrush. Thrush occurs a lot in children under 2 years old. Why?
First is the weak.
Yes, because yes, it's because at that time a child will not have sufficient immune system in order to resist fungal infection, and that's why children below 2 years old normally get thrush. This person looks to be old, have trust, most likely have immunity. Okay? What is no? Yes, HIV. Why does HIV lead to a lot of fungal infection?
That's suppressed immunity.
They don't get suppressed.
I mean, like, yeah.
They're not, yeah, they are immunocompromised because HIV kills CD4 T cells. And when CD4 T cells are gone down the drain, you become weak impact. Alright? And therefore, HIV has got one of the highest rate, those HIV patients have one of the higher rate of fungal infection. And their fungal infection are quite deadly as well. Okay? So, what is the innate immunity towards fungus? Well, the principal mediator of innate immunity against fungi is neutrophils. So, what neutrophil does is that they liberate fungicidal substances such as reactive oxygen species, ROS, okay, as well as lysozyme enzymes, okay, which can break down fungus. They also phagocytosize fungi for intracellular killing. Okay? And in terms of acquired immunity towards fungus, it's normally cell-mediated immunity, which is the major defense. And then fungi that are present intracellularly in the macrophages are eliminated by the same cellular mechanisms that are effective against intracellular bacteria. So it's pretty much similar, whereby you need all your different. So they cannot escape. There's no way for them to escape. Why? Because they are a little bit too high up the order of what do you call that now? Yeah, on the taxa they're a little bit higher order. They are not your standard single cell organism. Okay? So basically they are a bit slow if they want to change their antigens or mutate and all those things. Okay? Next, parasite. Parasite, in infectious diseases terminology, parasitic infections refer to infection with animal parasites such as protozoa, helminth, and ectoparasites. This one you guys will learn a bit more in PFN 501 with Dr. Nurul and Dr. Shahida. Okay? Now, humans are only part of its complex life circle. Okay? Which means that there is only a certain part of their life that they are in the human. Because they could be in the environment, they could be in the soil. So there are soil transmitted diseases, there are zoonotic helminths, for example, from cats. I think it's what, spondylitis or something like that. It basically jumps from animals to cat to you through the feces and everything. Alright? So all of these are known as zoonotic disease because we are just part of its host. Okay? So in terms of parasites, protozoa and helminthic parasites that enter the bloodstream or tissue are often able to survive and replicate because they are able to resist the host's innate immune response. So parasites in human hosts, sorry not human, in human hosts are usually resistant to complement and macro resistant to complement and also because they have something called a tegument. Okay? So what is a tegument? The name is derived from the Latin word tegumentum or tegere, which means to cover. It is a characteristic of all flatworms, including the broad groups of tapeworm and flukes. So what it is that it's a syncytium, okay? A single cell or a cytoplasmic mass that contains several nuclei formed by the fusion of cell or by the division of nuclei, and it consisted of multi-nucleated tissue with no distinct cell boundaries, and its thickness is 7 to 6 micron. So what it is that imagine your phone having your phone cover. When you drop it, you don't break it. So for the parasites, they also have this sort of covers. How it's made up of is basically mass of cells because they are not single cell organism. Okay, they are multi-cell organism. So what it is that all the old cells that is being built up is then used as and because of that, they are really built to sort of have a little skin armor outside of them. Okay, imagine the thickness is 7 to 16 micron. Considering for a tapeworm is relatively thick enough. Okay, so you can see this is an example of parasitic infection. You got things like whipworm, tapeworm, roundworm, gladia, hookworm, and coccidia. Okay, so then what are the acquired immunity towards parasites? Because in terms of innate, there is some, but it's not really good. Okay, so the acquired immunity towards parasites are basically quite different. Okay. Different parasites elicit quite distinct specific immune response. So cell-mediated immunity is the principal defense against protozoa that survive within macrophages. Okay, but protozoa these protozoa can replicate inside the cell and lyse the whole cell to stimulate specific CTL or cytotoxic T cell response. Basically, evolutionary adaptation gives parasites the ability to evade and resist the immune response. Okay, some parasites survive and replicate inside cells, while others develop cysts that are very resistant to human response, immune response. Sorry. And parasites can develop integument that is resistant to damage by antibodies and complement or CTL. Okay, so some parasites also have mechanism for surface antigen variation, so they can also have some changes. And there are also two forms of antigenic variation, whereby it's very stage specific, or they could basically have continuous variation of the major surface antigen. Why? It's because, for example, a helminthic infection, you have the nematodes, you have the I can't remember. There's so many. Yeah, I think so. I can't remember very well. I understand a bit, but not as well. But just wait for Dr. Noro to give you all the class on the parasitic infection. Okay? So the thing is that because of the variation in the stages and everything, once they change form from A to B, then they will have changes in terms of the surface antigens as well. So it's a bit hard to get rid of. Okay? Imagine, can you recognize if you give somebody who haven't eaten egg before, an egg with the shell, a hard-boiled egg, and also a fried egg? You can't recognize that all three of them are the same, right? If the person haven't seen an egg before. It's that sort of a situation whereby it depends on at what stage you can try to get rid of what antigens. Okay? That's why vaccines, again, parasitic disease are actually very difficult to develop because you don't know at which stage you need to target them. Okay? It's just too difficult. Okay? So these are an example. Okay? The evasion mechanism. So you got your antigenic variation, evasion of macrophage, resistance to complement, immune suppression, surface and secreted antioxidants. Okay? And those are examples of the parasites. Okay? Okay. So don't worry about memorizing all these. Don't go and remember what are the parasites, what are their mechanisms. Just understand the mechanisms will be sufficient. Alright? Yes. Okay. So yeah. So I'm done with infectious diseases. Okay. So a little recap. Not all diseases are contagious. So this is for non-infectious disease. Not all diseases are contagious. A disease that is not contagious is called infectious disease. These diseases are not caused by pathogens and blah blah blah. So the common type of non-infectious disease include cancer, diabetes, autoimmune disorders. Okay. So, okay, I need to break. Yes. Take a 5-minute break, people, and then we'll continue with cancer and immunity. Alright? I think I'm more than halfway already. Yeah, I'm more than halfway. So yeah, take a 5-minute break. Correct. Yes. So in Chinese, you got fu. Okay. So yeah, it's a variation. So different people have different levels of acceptance. But if you want to take beans, the fermented ones are the best because the toxins are being gotten rid of. Okay, I'll be back in 5 minutes. Which even though the antigen unit find the cells with it. Why? Because most of the time it basically loses out its antigen. And when it loses out the antigen, what's going to happen is that you cannot recognize it. Alright? You also have tumor-induced suppression. This is where things like TGF beta, okay, which is what you call it? A suppressive cytokine is being produced. Okay, imagine you have a cancer cell, you have a T cell. Okay, the T cell is trying to attack the cancer cell. The immune system. So be it if it's an antibody or be it B cell, you can't actually infiltrate it. Okay. You also have tumors with fast ligand. Okay, which basically stops the tumor from basically stops the T cells from attacking. You also have things now like PD-1 and PD-L1. Okay, so PD-1 is the programmed death 1 receptor. Okay, and PD-L1 is the ligand. Okay. So what's going to happen is that on all our cells, there is a PD-1, the programmed death. Okay? But the thing is that normally you don't have PD-L1 so much on normal cells. Okay? But in cancer cells, they upregulate PD-L1. When T cells come into contact, they force the T cells to die through PD-1 stimulation. Okay? So that's also one method of this one. And lastly, not in the graphic, is regulatory T cells infiltration. Okay, so regulatory T cells happens when you have a persistent or a chronic expression of a certain antigen. Okay, no matter how the immune system try to get rid of it, okay, it's basically we're supposed to be providing things like IL-2 and interferon gamma and all those things, is then converted into regulatory T cells, okay, which then expresses or excretes things like TGF beta, IL-10. Okay, and when that happens, your T cell will go into a phase whereby it's being suppressed. So that's why it's called regulatory T cells. And normally this whole thing is called regulatory cell suppression. Okay, so 7, I think. Somebody called Sakaguchi. So it's a Japanese person. Alright? Treg, E-R-E-G, or regulatory T cells. Okay? Right? So these are the mechanism of tumor infection. So that's for cancer. Now in terms of diabetes, diabetes is another type of non-infectious disease. Now diabetes occurs when the pancreas does not make enough insulin, or else the body cells are resistant to the effects of insulin. Now, insulin is a hormone that helps cells absorb glucose from the blood. Okay, so you've got your type 1 and type 2. Let's see if this is active. Okay.
No. The videos are available.
Wow, video not available here. Never mind. Basically, it's a simple this one that tells you more about type 1 and type 2 diabetes. Produce insulin, okay, and the warning signs are that you have increased thirst and urination, constant hunger, weight loss, blurred vision, and extreme tiredness. Now, type 1 diabetes is caused by genetic or environmental and autoimmune factors. Therefore, it cannot be prevented. Okay, so type 1 will normally go on with it, right? And the treatment will normally be insulin injection, oral medication, diet, exercise, and glucose monitoring. Okay. While type 2 diabetes is basically either that the body does not produce enough insulin or the body does not react to insulin anymore. And the early signs are that you are fatigued, you have slow wound healing, pain and numbness in hand, feet, and symptoms can be mild and go unnoticed. Okay, it's normally caused by lifestyle. Okay, lifestyle, genetic, aging, and therefore it can be prevented if a healthy lifestyle, including eating sensible and exercise, is being employed. Okay, treatment basically diet and exercise. That's the most important one. But if in terms of very severe cases, insulin injections and also oral medication are also prescribed. All right.
But this doesn't effect. It's not so much effective for those who are body.
Yeah, true, correct. So that's why you still need other oral medication. So it's just not insulin itself. You also have other medication involved. So if you have anybody who has diabetes, they go through other medication first, and then insulin normally the last line. Why? Because they don't want to oversensitize insulin in the body. Alright? Now, type 1 diabetes is more related to immunity. Okay? It is because the immune cells attack and destroy the pancreatic cells, and the cells can no longer produce insulin. This type of diabetes occurs usually develops in childhood or adolescence, and there's no known way to prevent the development of type 1 diabetes. However, it is still a treatable disease. And the treatment of type 1 diabetes includes taking several insulin injections every day. Interesting part, immune system diseases, right? So the immune system is the body's system that normally fights an infection and defends against it. But when the immune system is not working well, it usually keeps you from, when the immune system is working well, it keeps you from getting sick. But the immune system can also have problems and develop disease. The two main types of immune disease are number 1, autoimmune, and number 2, allergy. Alright? So anybody here has got allergy?
Yes.
What allergy you have?
Alcohol.
That's not an allergy, no?
That's very sad.
That's not an allergy. You get like rashes and then you can't breathe after you take alcohol.
I get rashes.
You get rashes, that's just a minor allergy. People who have peanut allergy, that's terrible. Because once they take peanut, they can't breathe. They got constriction of the trachea and everything. Small problem.
Small problem.
Yeah. Small problems.
Sure.
Alcohol is not the end all and be all. Okay. Now, in terms of autoimmune disease, okay, it is a disease whereby the immune system attacks the body's own cell. Now, I explained to everybody that how you actually got all the cells that attacks your own cells out of the way, right? So by right, there shouldn't be any more autoimmune disease. Okay. However, autoimmune disease still will occur. Okay, as we develop. Okay, there are multiple causes. Okay, something that I'll cover in our autoimmune lecture later on. But there are multiple factors, and actually autoimmune disease is the most difficult disease to treat. Okay, you can say that cancer is very difficult to treat. Okay, but there are still specific drugs. The problem with autoimmune disease is that it's so broad that it's not only caused by a single factor or two factors. It could be caused by up to 15, 20 different factors. Okay? For example, I have psoriasis, it's a skin disease. Okay? Why I got psoriasis and why other person got psoriasis might also be different cause. Okay? Why? Because it's autoimmune. Right? It depends on the body's immune system, and it depends on the environmental condition that the person has gone through. Okay? Yeah, because of the high dose of penicillin, so I lose quite a big number of my blood micro. And when that happens, certain food that I take might actually cause a problem. It could maybe be as simple as just flour. Yeah, rice flour.
So
Or maybe
you
wheat
didn't
flour.
have the psoriasis before?
No, I didn't have psoriasis until I was 18 years old. That was when I had the bad infection, high dose of penicillin. That is when I know that the gut microbiome would have gone down. So it could be various factors, right? Stress. I need to be performing to my absolute highest level anyway. You'll be surprised. I receive scholarships for all my education, so I don't need to pay for my education at all. I don't pay a single cent for education at all. So yeah, I was supposed to go to MIT after my Form 5.
Okay.
Supposed to go, but the thing is that I told them I don't want to be an engineer. I want to be a doctor. So then they said, nah, it's either you be an engineer or you want to go medicine. That's fine. I'll just take my chances. So yeah, and then I ended up in Form 6, and then didn't go into medical, but I ended up here. Anyway, that's the past. So so that's how autoimmune is. Okay. So one example is also type 1 diabetes. Okay, whereby it's an autoimmune disease. In this case, the immune system attacks the cells of the pancreas. And other examples are multiple sclerosis and rheumatoid arthritis. So remember, like some Chinese people, there's just a few Chinese people where you have Hong Xip, Hong Sap. Okay? So that is actually rheumatoid arthritis. It's not caused by bloody weather. It's actually an autoimmune disease. Okay? So if for those people who don't know the Chinese culture, Chinese people, when they get older, they get rheumatoid arthritis. Okay, so they call it Hong Sap. So it's wind cold, wind wet. So it means it's very damp. So it causes their joints to be painful. Alright? So they always blame it on the weather, but actually it's just.
Well, those are habits. They say they cannot sleep after you just shower.
Oh, that's a different story. That's a different story altogether. But yeah, that is Chinese culture, alright? But actually it's just rheumatoid arthritis, alright? So in multiple sclerosis, the immune system attacks the nerve cells, causing weakness and pains that gradually gets worse over time. And in rheumatoid arthritis, the immune system attacks the joint. This causes joint damage and pain. So these diseases can't be prevented and have no known cure. However, they can be treated with medicines that weakens the immune system's attack of normal cells. So if I want my psoriasis to go down and look to have perfect skin, the only way I can do is I can take immune suppressions. So I used to even take methotrexate, which is a drug for treating cancer. Okay? So people who take it to treat cancer, they take it on a daily basis. I take it on a weekly basis. Okay? Why? Because it targets fast dividing cells. So The immune cells are fast dividing cells, the skin cells are fast dividing cells. So when you take that, it sort of slows them down. When it slows them down, I don't get the symptoms on my skin. Okay, so I look fairer. Substance as though it was a pathogen. So any substance that causes an allergy is called an allergen. And the most common allergens are pollen, dust mites, mold, animal dander, insect stings, latex, and certain foods and medication. Alright, so that is allergy. Now the thing with allergy is that you have severe cases of allergy or you have mild rashes, like him. Okay, so the more severe ones are the peanuts and etc. There are also people who are allergic to sort of different medication. Okay, like myself, I'm allergic to sulfur-based antibiotics, which has been phased out. Okay, so last time when I was younger, during the 80s, sulfur-based antibiotics were still used. In terms of that manner. Okay? So it's all very dependent. Okay? Anybody has got hay fever? Hay fever. During summer, you get sneezing. No, not summer, spring. Spring. What? No. Spring? No. So during springtime, there's a lot of pollen floating around. So people get hay fever. So it's quite serious, whereby some people will be off for a couple of days. It's basically non-stop runny nose, fever, sore throat, and etc. Okay, so those are allergy.
So what? Is it Western?
I'm not too sure, but there are also, well, if you say peanut allergy happens to Westerners, people in Asian countries used to have lactose intolerant. Yeah, lactose intolerant. Why? Because there's a possibility that we don't take that much milk. Okay, so Asian population we tend to take a lot more other milk, soy milk, almond milk, and all those stuff of milk. I think goat milk, right? Yeah, Asian goat milk. So yeah, so sometimes Asians develop lactose intolerant, Western people develop peanut allergy. Yeah, so it's very much dependent. Okay.
I think it's more common to see like those Western restaurants have like, oh, this is peanut free.
Yeah.
You can't see those like, oh, we indicate it's peanut free.
Yeah.
You don't give a shit about this thing.
Yeah. Everything is got milk. Okay. So I think it's dependent on our ancestral diet, and I think during the separation of the continents, where different people grew up in different places, the environmental factor basically causes a sort of genetic pressure, and that pressure basically leads to selection of. So that's the end of the topic today. Any questions from anybody? So you notice I basically just go through the fundamentals of diseases, how the immune system actually is involved, okay, and then in terms of the in-depth of how these things happen and everything, you learn it later on with other lecturers. Alright? Because normally my class is at the second. Second half semester. But we just told them this year, just go on. So everything, so you get to attend all 4 different classes. Or only it's 2 classes and then 2 classes. But
It was more second part.
more second part.
Oh, sorry.
So towards the end.
Yeah,
you got one towards the end.
there's one is towards the end.
Because the thing is that imagine if you need to do my lab report towards the end of the semester.
Okay.
So then that will be headache. So that's why it's better if you spread it out a bit more. So I think this time you just do 3, 1. So the rest of the class are quite well spread out. Okay? Any other questions before we end? Do I have a class with you all today this week?
Not sure. Check.
You all learn how to do bleeding and then you come back, we keep the sample, we'll go and have lunch, then we meet up again for the second half of the experiment. Okay? So Monday will be quite a long day. Alright, so that's all. Thank you very much everybody.
There's still one slide after this.
You go back?
Yeah.
No more already?
This one.
Oh, this one was for the previous class. So this is last time. If you notice the date is 2022. So that's why this one I removed already. So don't bother with that. Just let it be. Okay?
Okay. Thank you, Doctor.
Thank you.
This should probably be with P.